Hello class, my name is Eric. I'm a student in Biology 3231, Biochemistry Part 2. And for my clinical case study presentation, I will be doing it in the format of a video presentation on this article. The article is called Final Diagnosis, a 2-year-old male with Fragile X syndrome. I found this article to be very dense and filled with scientific jargon. So the aim of this presentation is to break down the jargon and present the article into layman's terms that can be more easily digested by biochemistry students. So now I will start this presentation by providing a brief introduction to the syndrome and by incorporating information presented by Dr. Aurora in his introduction to this topic. The hallmark of Fragile X syndrome is mental retardation. Fragile X syndrome is the second most common genetic cause of mental retardation after Down syndrome. Mental retardation is defined as a non-progressive disorder evident during childhood with disabilities in adapting to the environment and an IQ score below 70. Prominent parental concerns that might bring a child with Fragile X syndrome to a pediatrician's attention very shortly after birth include developmental delay, speech delay, short attention span or hyperactivity, mouthing of objects persisting at an age beyond expected, difficulty in disciplining the child, frequent temper tantrums, autistic-like behaviors, and more. So, a result of the genetic cause of mental retardation is as follows. Those who are inflicted appear neurotypical in appearance. They appear ordinary or what you might consider normal. So consequently, people and professionals often mistake those who have this mental retardation as they might make the mistake that if you have a genetic cause, you must look different. So I just want to drive home a point of emphasis that this is not the case. This is a common misconception. So I'm going to try and digest this article by presenting to you first the big picture. The cause of Fragile X syndrome is a mutation on a specific gene called the FMR1 gene, which is located on the X chromosome. The loss of the FMR protein, which is the protein that is produced from this gene, the FMRP protein has loss of function. As a result, abnormalities both structurally and functionally in neurons result, and this reduces synaptic strength. Okay, the result of this is impaired learning ability. So first we discussed the mutation to the FMR1 gene. I wanted to expand on this piece a little bit more because this is of particular importance to biochemistry. So what happens is an expansion of the CGG trinucleotide repeat, okay? And these three nucleotides, this trinucleotide repeat, is located in the 5' untranslated region. So those who are neurotypical have 29 to 30 CGG repeats. Anywhere between 6 to 45 repeats is considered neurotypical. And typically we see 29 to 30, okay? So those with slightly above 46 to 55 repeats, this is a gray zone, right? Those in this zone are unaffected. They have a low risk of this expanding to be categorized as a premutation and an extremely low risk of expansion to a full mutation in distant generations, okay? You would say a premutation is 56 to 200 repeats of the CGG trinucleotide sequence, the same one that I've been discussing for the above two examples. Okay, in this premutation, DNA is not methylated here, and therefore the FMR1 gene is not silent. Premutations may expand to full mutations in future generations. Okay, and lastly, full mutations are considered a 200 plus repeats of, once again, the same CGG trinucleotide repeat. Okay. The next step after this, if you have a full mutation, is to proceed with methylation studies. So I know I just mentioned the term methylation studies. Allow me to elaborate a little bit more in the context of the diagnosis procedure of what this means. Diagnosis of Fragile X syndrome is done by a Southern blot analysis. Okay, Southern blot analysis tests DNA, and it actually identifies the size of the CGG repeats as well as the methylation status of the CPG island. So what happens is there is a restriction enzyme is used to determine if the region upstream of the FMR1 gene is methylated or not. An EcoR and methylation sensitive EAG are the enzymes that are used for those that are curious. 200 plus CGG repeats, like I said before, the full mutation, leads to extensive local methylation upstream from the repeat. So this is what you see. And the result of the methylation is transcription silencing, right? This is because transcription factors cannot bind. And due to the silencing of transcription, this leads to the loss of the FMR1 protein and clinical features that are characteristic of Fragile X syndrome. Okay, the two main outcomes of affected males in a full mutation, right, which leads to the loss of the FMR protein, and secondly, premutation, which increases transcription of the mutant FMR1 gene due to the lack of the normal protein, and accumulation of this abnormal transcript leads to the formation of neuronal internuclear inclusions. Okay, and this leads to the sequestration of important cellular proteins, which ultimately leads to cell death.